Okay, thanks a lot. Yeah, first of all, thanks to the organizing team for making this really nice meeting happening here today. I would like to introduce some recent work that we've been doing on learning the dynamics of cell-cell interactions in confined cell migration. And so in many of the physiological processes in which cell migration is important, ranging from the dynamics of epithelial sheets to developmental processes, but also tumor invasion during cancer metastasis. Cells constantly interact with one another, and they do so in such a way to organize these physiological processes. And the big question that we wanted to address in this work was whether there might be simple dynamical laws that describe the emergent long time scale behavior of interacting cells. And to address that question, we tried to build really a minimal sort of biophysical setup in which we can monitor cells colliding with each other, which is much, much simpler than these physiological processes I'm showing here. And the system that we came up with is this two-state micropattern where we have two square adhesive sites connected by a thin bridge. And what happens when we put pairs of breast cancer cells into these micropatterns is that they repeatedly collide with one another. And sometimes they even manage to make a transition and actually move past each other. And so this system is sort of like a minimal cell collider where we can place cells into this very standardized environment and then watch them collide over and over again. And the kind of data that we're collecting here is our trajectories of the cell nucleus. So we label these nuclei and then track their trajectories. And by looking at many of these micropatterns in parallel, we can gather a huge data set of coupled cell migration trajectory, of which I'm showing you a small subset here. And we did this for two different cell types. One's this non-cancerous MCF10A breast tissue cell, and for a cancerous breast tissue cell. And the first thing that we did when we had collected this data was to see in the videos and the trajectories if there are sort of stereotypical repeating behaviors that we see over and over again. And what we notice is that when these cells collide, they quite often perform something that we call a reversal event, where they essentially collide and then turn around upon collision. Sometimes we also see that these cells collide and then actually interchange positions and therefore slide past each other, which we call the sliding event. And the third sort of collective event that we found was where cells actually stick together and then make joint transitions, which is something that we called following. And now what's interesting is that for these non-cancerous cells, we found that they predominantly reverse upon collision. And that's actually a behavior that's been identified previously in the biological literature and been termed contact inhibition of locomotion, which is a behavior that's been observed for many different types of cells. But what was surprising to us was that when we applied the same analysis to the cancerous cells, we actually find that they preferentially slide past each other upon collision. And so they don't seem to have this kind of contact inhibition of locomotion behavior. And so the question here is, given that we have these two cell types with very different sort of behavioral phenotypes, is there still a unifying quantitative framework for the dynamics of these distinct behaviors? And so our aim is sort of to derive a quantitative description of these long time scale stochastic trajectories that we observe in these videos. And the way that we think is a mathematically useful way to do that is using stochastic processes. And this has actually been known for a long time for the migration of just single isolated cells on unstructured isotropic environments, like I'm showing here in this video, where these cells perform some kind of random walk. And you can describe their trajectories very accurately through a stochastic differential equation, which is basically an equation of motion for the cell, which gives its acceleration as the sum of a deterministic persistence term and a stochastic Gaussian white noise term. And in previous work, we actually generalized this type of description to the problem of confined single cells. So here we looked at single cells in the same confinement, where we were interested in how these cells managed to overcome this thin constriction in the middle here. And what we found for the dynamics of single cells is that we can actually generalize this equation of motion framework, where we now have an average term that also depends on the position of the cell since it's migrating in a structured environment. And what we now thought that we can essentially generalize this framework that we have for single cells to the problem of interacting cells. And so our intuition here is that we can again write down an equation of motion for these cells, which additionally to a single cell motility term might also have interaction terms, which in this case we postulate take the form of a cohesive interaction, which could range from repulsion to attraction, and a frictional interaction, which can depend on the relative velocity of these cells. And what we now do is we use the trajectories that we observed experimentally, and we use the short timescale information in these trajectories, so the local accelerations, to infer these distinct terms of this equation of motion. And so first, for the single cell term, we find this dependence here, which actually looks extremely similar to what we found for experiments where we really only had a single cell in this confinement, showing that the single cell dynamics don't change much when you add another cell. But we can now also infer the interactive terms of these dynamics, which include a repulsive interaction and a frictional interaction where the cells slide past each other and then slow down. What was interesting to us is when we did the same analysis for the cancerous cells, we found a similar single cell term, but really qualitatively different interaction terms, where we had now an attractive interaction and sort of a novel and surprising anti-friction interaction, which actually means that when cells slide past each other, they speed up upon collision. Now, we shouldn't just blindly believe this model. We actually have to test whether this model can capture our data. And to do that, we simulate trajectories. And remember here, we only use short timescale information to infer this model, but we now actually test this model whether it can predict long timescale features of our dynamics, such as these reversal and sliding behaviors. And we find that the model actually captures the ratios of these behaviors very well. So this shows us that the general form of this equation of motion that we've postulated here can accurately capture the dynamics that we see in experiment. But we now wanted to try and sort of generalize this framework that we've built here beyond the two specific cell types that we observed in this experiment. And so to do that, we realized that we can actually draw sort of a two-dimensional phase space spanned by the cohesive component, which can range from attraction to repulsion, and independently the frictional component, which can be anything from friction to anti-friction. And based on our inference, we already know that the cancerous cells should be somewhere up here where we have a combination of anti-friction and attraction, and the non-cancerous cells down here where we have repulsion and friction. But now we wanted to extrapolate beyond these two data points that we sampled in our experiments. And so we could actually run simulations with, for example, combining a frictional and a repulsive interaction. And in all these cases, we find that reversal events dominate this entire region here. And so we call this sort of a contact inhibition of locomotion behavior. However, if we put in an anti-friction into our model, regardless of the parameters, we always find that the sliding events dominate. And we call this contact sliding locomotion, which is also in line with the fact that we found that the cancerous cells are somewhere up here. Finally, if we combine attraction and friction interactions, we found that the cells actually stick together most of the time, which is not something that we saw with the two cell types we have here. And these cells actually make joint transitions, and we call this contact following locomotion, which is actually a type of interaction that has been observed in other systems before with different cell types. And so to summarize, what this shows is that our model can not only just capture these two cell types, but it's actually sort of predictive for these distinct different behaviors that you can see in a range of different biological contexts. And with this, I would just briefly like to summarize the general philosophy of this approach, which is that we can access these stochastic trajectories in our experiments, and we no longer have to guess what are the what is a good model for these trajectories, but we can actually infer that directly from the experimental data, giving us access to these long timescale dynamics of the system. But the hope is now that we can use these dynamics to somehow constrain the underlying mechanism that drives this migration. And on the other hand of the scale, also connect this to the behavioral scale, where we look at long timescale behaviors of these cells, such as these collision events here. And with this, I would like to finish and thank my collaborators, especially Alexandra, who performed all of these experiments in the lab of Joachim Redler, and also my advisor, Chase British at the LMU Munich, and all of you for your attention. Thanks.
Thank you, David. Really nice talk. So we welcome questions now.
I have a question.
Mm-hmm.
I don't know if I was the first one though. But
just go ahead.
Yes, just go ahead.
So really cool talk and really cool work.
Thanks.
So I have several questions, but I guess I'll try to focus on two small ones. So the first is your single particle term that you infer looks very complex. So I was wondering whether you tried, for example, different confinement shapes, whether this was due to the confinement box that you used, or what made it complex in general. And the second question that I had, actually I forgot. Yes, the second question was if the interaction terms, you told us that there are different interaction modes, but the interaction terms that you infer are integrated in time. So in a single, let's say, movie, could there be different interaction modes that you integrate basically on in your inference? Sometimes they stick and they move coherently, sometimes they repair. So that's the question.
Yeah, thanks. Yeah, so regarding the first question, essentially what is the nature of the single cell dynamics? So that's essentially the subject of this paper here, and I probably won't have time to go into all the details, but essentially we do need that complex nonlinear dependence that I showed here, and what you can see is that this is essentially the position of the cell, and as it makes a transition from the left hand side to the right hand side, it's essentially accelerating. And we found that you can describe this essentially as some kind of nonlinear oscillator that is speeding up as it makes this transition. That is sort of the gist of this. And we actually did look at a lot of different types of geometries. And basically what we found is that what is important here is not the exact shape of these islands and so on, but the fact that there is this transition here. And so if you make this transition extremely broad, then this feature that the cells speed up disappears. But as I say, all the details are here in this paper. And regarding the second question, you're absolutely right. We only consider time stationary dynamics here. So that's essentially the first assumption that we make here, that we don't have time-dependent dynamics. And you might be able to fit different models that do depend on time, but we would argue that a time-independent model is a simpler thing to try first, and we find that this captures our statistics. And so we did not consider time-dependent dynamics.
Okay, thanks.
Yeah.
There is a question by Lorenzo.
Yes. Can you hear me?
Yep.
Okay, first of all, I would like to thank you for your very nice talk. And my question just regards the fact that looking at your single trajectory and at the model you considered, basically it seems that you are just assuming a sort of persistence in the dynamics, making an inertial term. That's right?
Yeah, that's right. Yeah.
Okay. So this is basically looks like very similar to model in the field of active matter, such as the active Brownian particles model or the active Austen-Uhlenbeck particles model. That's actually basically coincide with the active Austen-Uhlenbeck model. I'm just wondering if the same kind of strategy could basically be adopted. So for the case, so for the confinement case, since of course when you play with interaction terms, some differences could arise if you are assuming or not. So the fact that the active Austin-Uhlenbeck and the in the underdone model with a frictional force are the same is just a consequence of the absence of interaction. While instead, when you consider interactions in the model dynamics, you can clearly see some differences. And I'm wondering if you have already considered this aspect in the
that's a good question. So just regarding essentially the aspect for the single cells, you're completely right that here we assume underdamped dynamics. So we have essentially an effective inertia, which is why we describe the acceleration of these cells. And so the 2 degrees of freedom that we consider here are the position and the velocity of the cells. And you're right that you could rewrite this equation of motion as essentially an overdamped equation. So a first order equation of motion for the cell position, plus some time correlated polarity, right? Which could be your active Ornstein-Uhlenbeck process that you were referring to, I think. And that could also be a valid description of these dynamics. But the problem is that we don't observe that polarity, right? We don't have a simple experimental observable for what is the polarity of the cell. But we are working on essentially deriving a simple mechanistic model that would include such a polarity, which could then reduce to this kind of equation of motion here.
Okay, but your terms that you write as a f that is a function of x and v. I guess that in this term x and v are coupled. That's right.
Yeah.
So basically, you are just looking at a non-equilibrium dynamics where these 2 terms come somehow together, and these non-equilibrium terms. So I don't know what is your choice of f, since I can see here, but I know that there are some backward mapping to this choice to something like that looks like similar
maybe it's possible to keep discussing that in the coffee break, since maybe
yes, I'm really sorry.
Sorry for the long question.
No
yes,
worries.
there are other questions, but there will be a coffee break in half an hour. So if you want to talk with David, it's going to be the opportunity. Okay, thank you very much, David, and see you later.